Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between the Associate Editor-in-Chief of Molecular Therapy Advances, Dr. Dan Stone, and Dr. Dan DuBreuil from Sanofi. They will discuss an article recently published in Molecular Therapy Advances titled Development of a Secretable Frataxin for Enhanced Efficacy in Treating Friedrich's Ataxia. Before we get started, ASGCT is hosting a one-day graduate medical education boot camp in cell and gene therapy, perfect for clinical fellows, residents, advanced practice providers, junior faculty, and early career investigators. Learn how to safely and effectively integrate cell and gene therapies into patient care. Join us on August 21st at Nationwide Children's Hospital in Columbus, Ohio. Visit asgct.org to learn more. And now I'll pass it off to Dr. Dan Stone.
Thank you, Rory. Hello and welcome to the Molecular Therapy Podcast. My name is Daniel Stone, your host for this episode, and I am the Editor-in-Chief of the ASGCT journal Molecular Therapy Advances. Today we're recording from the ASGCT annual meeting in Boston, and we will be discussing a paper that was published this year in Molecular Therapy Advances titled Development of a Secretable Frataxin for Enhanced Efficacy in Treating Friedrich's Ataxia, which describes the development of a novel approach to treat Friedrich's Ataxia. Friedrich's ataxia is a progressive disease caused by trinucleotide repeat expansion within the first intron of the frataxin gene that causes loss of frataxin expression, leading to mitochondrial dysfunction and toxic accumulation of iron. This in turn causes neuronal degeneration, dysfunction, scoliosis, and arrhythmia due to hypertrophic cardiomyopathy. It's one of the most common inherited ataxia with a prevalence of about 1 in 50,000 people. Although onset of the disease may vary, typically symptoms appear between ages 5 to 15, and its progressive nature means that patients often become wheelchair-bound by the mid-20s, and heart complications can cause premature death. The paper we will discuss today offers a potential single-shot treatment for Friedrich's ataxia via delivery of a modified secretable frataxin protein following administration of an AAV vector to the CSF. The authors demonstrate system-wide delivery of frataxin to impacted organs such as heart, brain, and dorsal root ganglia in small and large animal models. They also demonstrate rescue of neuronal and heart phenotypes in a mouse model of Friedrich's ataxia. This work was performed at Sanofi, and today I will be joined by first author Daniel DuBreuil, who it's my pleasure to introduce. Daniel received his bachelor's degree from Otterbein University in Ohio, his PhD at Brown University in the lab of Diane Lipscomb, and did a postdoc with Brian Wagner at Mass General in Boston. Daniel, it's a pleasure to have you with us today.
Thank you for having me.
So I thought I'd get us started by asking you an overview question. So Friedrich's ataxia seems to be a very highly complex disease. I was really learning about it a lot this week, didn't know a ton about it, and it impacts multiple different organs. So what are really the main challenges for developing a potential treatment or cure for such a complicated disease?
Yeah, so that's a great question. It's one of the challenges that we thought about as we were, embarking on this endeavor. And it really comes down to the key tissues that need to be treated being at such different parts in the body, having those dorsal root ganglion neurons and neurons deep in the cerebellum, and then balancing the need to treat those cells and restore frataxin there with the heart to really correct that lifespan challenge that posed the real challenge. And so that's what we were kind of focused on when we were thinking about what our strategy would be.
Yeah, that's very interesting you bring that up, how you've got the different organs in different regions. And that kind of brings me to my first question here. So in our lab, we're targeting dorsal root ganglion, but from a perspective of an infectious disease virus, but we're not looking at the same organs like the heart, maybe somewhat the CNS for you. So I see you selected to use AAV as your delivery system, as a lot of people are doing in similar, and you're using an engineered capsid that I believe was developed in Beverly Davidson's lab.
Yep.
Now it seems to transduce CNS and PNS very efficiently in heart. Was this variant initially selected in NHP or in mice, and how were these selections performed? Is it more specific, or is it just very good at transducing everything?
Yeah, so this was originally screened and identified in non-human primate. So we knew we got very good biodistribution in primates. And it was initially kind of developed through a series of different variants that we were evaluating. And then this one really stood out to us because it had very high expression levels from low vector genome copies. So we knew that we could get broad biodistribution, but also that we didn't need to get a ton of vector genome in each cell to get the levels of expression that we would need. So that's what really made it stand out to us.
Yeah. No, I wasn't actually familiar with that particular. I mean, there's so many different variants, right?
Yes.
That people. I wasn't familiar with that one, so I had to do a bit of background reading on that. So it was very interesting. Some of the early work in the paper, you're basically optimizing all the different parts of your vectors and your treatment. Totally makes sense. You decided to go for a tissue-specific promoter-based approach, initially looking at synapsin and then this new Desmond promoters.
Yeah.
Didn't seem to go quite how you'd expected. The experiments on that was quite interesting. Specifically the new Desmond promoter, which is cardiomyocyte specific or not, as it would case. Do you have any insights as to why it expresses beyond muscle, in CNS and PNS, and it seems to be in liver and heart as well, which you wouldn't necessarily predict?
Yeah, I think it's a really great question. I think it really comes down to the size of the promoter and the different regulatory elements that we're able to include there, and just that we're not capturing all of the regulatory elements that would be necessary to really refine that expression profile to match what the Desmond gene is actually doing endogenously. And so just that combination of factors seems to drive enriched expression in skeletal and cardiac muscle, but not the level of specificity that we might have anticipated. But that's actually a great benefit to us to get that kind of low efficiency expression in these other tissues.
Yeah, so one thing that other people have shown is when they're using tissue-specific promoters that can kind of reduce host immune responses.
Yeah.
I know you don't really go into that in the paper much, but have you kind of looked as to whether the leaky expression is impacting host responses, or you didn't really look at that?
Yeah, it's a great question. We haven't looked at that here. It's something that we definitely thought about doing as we kind of move forward, but so far we haven't had a chance to dig into that.
Sure. And then one kind of final question about the Desmond stuff. So you also, interestingly, show that when you have monocistronic versus a bicistronic vector, that you get reduced expression with the bicistronic vector.
Yeah.
So it's been shown that having repeat sequences or inverted sequences can destabilize AVA genomes. So I was wondering, I know you did some codon shuffling in your different Desmonds in there, but there's probably still some similarity. I was wondering if you looked just to see if that impacted genomes, did any long-read sequencing or anything?
Yeah, it's a really great question. We didn't do on these particular vectors any of the long-read sequencing work to look at that vector integrity, but it's a really good point and definitely something that we were relieved that we could get away from to kind of get rid of some of those challenges associated with any repeat sequences or any folding of the genome or truncations and things like that.
Yeah, I know. We've had some issues with that in our lab, so it's definitely something.
Definitely going to be a big challenge for things moving forward.
I was wondering if you could talk us through the different approaches you used for the peptide optimization. You did some kind of AI stuff and some other predicted stuff, because you obviously want to get out and then back in and then go to the mitochondria. So you've got like 3 different aspects you're looking at there.
Yeah, so it was a really interesting problem to try to tackle, and it's really great that we had some of these in silico tools that we could rely on to make predictions. What made doing the optimization and the engineering so challenging is that we can't actually use in vitro models because they're not necessarily predictive of in vivo secretion efficiency and things like that. And so we really had to use a lot of in silico screening to design candidates and then move forward in vivo. And so we really, what I tried to do with the engineering was start with a broad panel that gave kind of some level of sequence diversity that then I could use to pick and choose pieces from the N-terminal or the C-terminal parts of these signal peptides and then incorporate them into the endogenous sequences in an iterative fashion to try to make them perform a little bit more efficiently.
Yeah, it's always tough when you kind of got to go straight to in vivo so you want to kind of minimize the number of things you're comparing. So that was very helpful. Thank you. So I wanted to kind of move on to the mouse work now. That's a kind of a good segue there. Now we talked about the complexity of Friedreich's ataxia a little bit just now, but to address, obviously no mouse model is perfect, but to address some of that complexity, you use 3 models, but 2 different mouse models. I was hoping you could just kind of explain to our audience just the differences between the mouse models and why you're using them and the limitations a little bit.
Yeah. So there are several different mouse models that are used for Friedreich's ataxia. Some that try to recapitulate the actual human condition, where they insert these trinucleotide repeats, but the behavioral phenotypes in those mouse strains are a little bit less clear and a little bit less robust than in some more artificial strains. And so what we decided to use were tissue-specific knockout lines that remove frataxin specifically from either the cardiomyocytes and skeletal muscle or from the neurons that are involved in the behavior. And that way we could get really good, clear behavioral effects that we could look at as kind of downstream readouts that we were providing, fully functional frataxin.
And then in the neuronal, the PV albumin frataxin mouse, does that knock out frataxin in both the CNS and the PNS?
Yeah, so it'll be subsets of neurons in both the CNS and PNS, but it hits the relevant cell types in the cerebellum and in the dorsal ganglia, which is great.
Perfect. Okay, I wanted to ask you about, so you get some really nice results in the neuronal model. Yeah. You show that you're able to rescue the ataxia phenotype both through neurological assessment and through the rotarod, which is very impressive. But not only do you see that with your modified, but you see that with the wild-type frataxin as well. So just delivery itself seems to be having some impact.
Yep.
In this particular step, you actually see minimal impact of the cross-correction.
Yeah.
Why do you think that is comparatively? Or do you just think you've reached close to maximal effect there?
I think that's a great question. And the way that we're thinking about that is with the route of administration and the capsid that we're using, we know we have really good biodistribution to those key neurons that are required for rescue of this behavior. And so we think that because we're getting our vector genome into the cells that we're supposed to be targeting, we don't need to cross-correct into them, so adding that engineering piece doesn't necessarily add a benefit. But what it is really great and showing us is that when we're having those cells actually secreting out the frataxin, we're still delivering sufficient quantities to have the therapeutic effect, which is something we were initially concerned about using these cells as factories.
Yeah, I think you did a really nice job of that. I want to compliment you on the quality of the ISH and the IHC images in the papers. You do a really nice job of showing that. So having spent a lot of my scientific career working on those things, I appreciate when it looks so nice.
Oh, I appreciate that.
So in the MCK frataxin model, the muscle-specific one, you see a small but significant extension in lifespan, but you only see this with the secreted ones versus the wild type one. And the effect, I have to say, it's minimal, but it's statistically significant.
Yeah.
So it's obviously a step there. Do you think that maybe this is just because you're treating the mice a bit too late? They're already 5 weeks old. Maybe the heart defects are already there, or maybe if you went for an earlier treatment, you might see a better effect there. Or do you have any other insights to that?
Yeah, I think that's definitely a possibility that we're treating a little bit on the later side. I think it's relevant to the patient population where they're going to have some level of heart dysfunction at the time of treatment. So I think that's at least a fair comparison. Yeah. What I think the biggest challenge is the difference between rodent and primate. Because this capsid was identified in primate, we know there are differences in biodistribution and function in a rodent model. And so really we see less biodistribution of this vector with our intra-CSF route of administration in these peripheral tissues in the rodent than we do in primate models. And so I think the level of transduction that we're getting is just a little bit lower than we would have liked to see.
Yeah, I also imagine be almost impossible to do an ICM and an IT injection on a mouse younger than 5 weeks old.
Yeah, that would be challenging.
Have you looked at I think if I remember correctly in reading some of the introduction, you've used systemic delivery and approaches. Have you tried looking earlier with a more systemic approach? Or
yeah, that's one of the things that we would really love to do is be able to get that dose response with a systemic administration and just see with our cross-correcting frataxin, how does our dose sensitivity look like, with that systemic route where we can really tailor how much vector genome biodistribution we're getting in the heart.
Okay, so I want to move on. I have a question about the NHP study. So in the NHP, the transduction of the heart was lower than in mice. Are you considering alternate capsids with better heart tropism, or do you like where you are with GM?
I think we like where we are with the GMU01 capsid. I think one of the things that, as everybody focuses on the heart, particularly with our route of administration, something that is underemphasized is the challenge in hitting those deep nuclei in the cerebellum.
Yep.
I think there have been several capsids that have been evaluated and tried, and they don't necessarily hit those correct cell types there. And so because we do actually hit those cell types in the primate, I think we're very comfortable with that, and just trying to figure out what dose gives us the level of transduction that we need to give us that footprint for the cross correction.
Yeah, it's an interesting question, because I was reading in your paper how you talk about patients who were questioned about this, and I went into the reference that you cite, are more interested in seeing an impact on the neurological functions than the heart functions. So that's obviously something you want to consider there. But at the same time, you want to be as good on both sides of the coin if you can.
Yep.
So it was really interesting.
Yeah.
It was very encouraging that you only see low-level neuronal toxicity in the DRG, and you didn't see it in the brain or spinal cord.
Yep.
That was performed in the and it was NHP.
That was in the primate.
That was in the primate.
Yep.
How does that dose compare to other studies where they've seen more advanced neurological damage?
All of our doses are allometrically scaled from what the maximum feasible clinical dose would be, and so this is a perfectly clinically relevant dose that we're using.
So that was really nice. And then this is probably what I thought was one of the most important parts of the paper in the discussion. So you estimated that from the scientific literature for Friedreich's ataxia that approximately a 60% repletion of frataxin expression in the heart would be enough to completely rescue, I want to reiterate that, heart-related symptoms, which is, a fantastic bar if you can reach that. Yeah. But actually 30% would provide durable benefits. Now, from your quite complicated analysis that I have to admit I didn't completely understand, but you appear to be pretty close to these levels across much of the brain, which is great. So you're quite close there. How can you get over the final hump?
Yeah, I think it's a really great question. This is something that we might think about, how is the dosing paradigm done? If you do slower or faster infusion rates, do you get more leak from the CSF into the peripheral bloodstream? Maybe there can be formulation changes that we can make that will, again, change that balance in biodistribution. So I think those are all things that we'll have to consider moving forward. But yeah, it was really close to, getting to those, full rescue levels. So we were really excited by that.
Well, so that brings me back to my next question. So now you've shown therapeutic efficacy in 2 mouse models, safety in NHP. Where's this going next?
Yeah, so that's what we're looking
into.
And
I realize you might not be able to share everything with us, but yeah.
Yeah, so we're looking into how we can move this forward. Like I said, there are some more studies that we want to complete in order to really round out this data package, get a full understanding of what sort of efficacy are we having in the heart with these different levels of expression and levels of biodistribution. And then we'll be looking forward to, how can we do our best to really get this moved towards patients and towards the clinic.
So that's obviously the aim for everyone, ideally. But, being in a company, I presume you're itching to kind of get into the clinic.
Yes.
Okay, so that was fantastic. So I think I've covered the main points I wanted to cover with the paper, but I'd like to finish by asking you something off topic. An idea that Rory gave me, and I always like to give her props for this every time. I understand you might not be able to share everything, but I wonder, are there any new and interesting projects or developments from your group that you're excited about and would be willing or able, might be the better, to share with us? It can be big picture, whatever you like.
Yeah, I think one thing that we're thinking about is definitely, this cross-correction idea is something that we've really thought about. How can we apply this in other ways to increase the efficacy of our different AAV or LNP-based therapeutics, right? And so we're looking into how can we take advantage of this for other indications, other sorts of, mechanisms that are separate from, direct repletion of a disease target gene.
Okay. Well, great. Well, Daniel, that was very useful. Thank you very much for joining us.
Thank you very much for having
Boston.
me.
That was very interesting. And we thank you, everyone, for joining us at the Molecular Therapy Podcast